And it's actually now time for our next speaker, which is Professor Gregg, Dean, who is a longtime collaborator of our favorites, our dear Todd Kleinhammer. He is currently the head of the departments of microbiology, immunology, and pathology at Colorado State University, and is working on mucosal immunology and vaccine development with lactobacillus acidophilus as his favorite microbe. So, Gregg, the floor is yours.
Thank you, Sarah. I appreciate that introduction, and thanks to the organizers for putting together this wonderful meeting and for inviting me to present this talk. Yeah, so this is an exciting session here. We heard so much about the positive aspects of probiotics and just their inherent value in terms of priming an individual for good health and response against potential pathogens and maybe prevention of disease. We're going to turn here in this talk to really thinking about how can we specifically target a pathogen like coronavirus by genetically engineering a probiotic as a vaccine platform. So really our goal is to develop a vaccine platform for use in low and middle income countries. So there's a fairly desperate need for such vaccines in these countries and these populations, environments, and there's some critical aspects that need to be addressed. So features that must be inherent in whatever vaccine strategy we look at. So first of all, it's got to be easy and inexpensive to manufacture. Optimally, it would not require a cold chain at all. So a room temperature vaccine would be incredibly valuable. Oral delivery, so that we avoid the need for a trained medical infrastructure to deliver a vaccine with needles. So being able to actually just hand vaccines out to individuals so they can take them orally on their own really would be a tremendous benefit in these environments. So in our lab, we're currently looking at rotavirus and HIV-1 in addition to coronaviruses, which is what we're going to focus on in this talk today. So, you know, the question that's kind of important to ask and address is, do we need another COVID vaccine? Clearly, there are a number of, excuse me, clearly there are a number of vaccines that are highly effective. They're being administered, and there is a global vaccination effort underway. So do we need any more? Well, we have to keep in mind that the vast majority of people have not yet been vaccinated, and that the risk of immune escape by new variants will be an ongoing concern. And I'll talk a little bit more about that. And if we think about that, and as we're attempting to vaccinate the global population, while we continue to have a high number of cases, we have this potential for variants to emerge and then require a booster vaccine. So then we end up in a catch up, always trying to get ahead of escape variants, which could be very difficult if we're looking at vaccinating globally, again, especially in these low and middle income countries. So we need a longer term strategy as well to prevent future coronavirus pandemics. And that's something we're very keen to address with a probiotic-based vaccine platform. So this is current data showing the percent of people who have received at least one dose of a COVID-19 vaccine. And if we look here at what's going on worldwide here at the end of May, only just over 10% of people worldwide have received a single vaccination. And we can look at where the pandemic is still really raging in South America and India and now Asia. and see that a fairly low percent of people have been vaccinated. So there's lots of opportunity for the virus to mutate and then for variants to spread fairly quickly and then become a threat to the vaccinated population if the existing vaccines aren't protective against that variant. So this is a big concern. So we are looking at lactobacillus acidophilus as a vaccine vector in particular, and we've been utilizing strain NCFM, which was isolated from a human by Todd Kleinhammer many years ago and has been widely used in a variety of consumables. And over this period of time, and as was mentioned in the previous talk, NCFM is generally regarded as safe by the FDA, and then we know that it has these health benefits, again, that were pointed out in the previous talk. Importantly, if we're talking about creating a recombinant bacteria, we need to have tools to manipulate it. We have those, and then inherent in the lactobacillus itself are these innate adjuvant effects. And really, this is what helps drive a lot of the beneficial immune outcomes. So I'm going to point out just a few expression strategies that we can use when we're thinking about delivering an antigen and an adjuvant with a bacterium. So with lactobacillus, we can look at surface layer associated proteins. And so up at the top here is electron micrographs of the bacteria with some cartoon overlays to give an idea of what the expression looks like. And then a cross section of the bacterial cell wall to also demonstrate how these recombinant proteins would be expressed. So they could be a fusion or a covalent association with the surface layer proteins. We can also insert short peptide sequences right into the surface layer protein. This is highly expressed, so we get abundant expression of whatever that peptide is, and I'll talk a little bit more about that in a minute. We can also express proteins in the cytosol, and we can secrete them. So we have all these options, and we choose these based on how we want to introduce these different recombinant proteins to the immune system. So I want to just step back for a minute and point out the real potential of using acidophilus as a vaccine platform, using, in this case, a model antigen, ovalbumin. It's a peptide that binds the mouse MHC2. It's a strong immunogen in mice, in C57 black 6 mice that we used here. And Bridget Eklund in my lab performed this study where she made this construct, she immunized mice, and beginning here at week 0 and then every 2 weeks thereafter to see what are the kinetics of induction of antibody at the mucosa. And so we're looking here at the fecal ova-specific IgA, and you can see at 2 weeks already all the animals are responding and have a high level of antigen-specific IgA, and then that's maintained throughout this whole immunization period. So the point being that we can very rapidly induce a mucosal immune response as well as a systemic response, which I'll show in just a minute. And so, you know, how is this happening? What is the immunologic basis for this? We can look at what we know about acidophilus and other lactic acid bacteria and how they engage specific innate immune receptors. And so shown here is the bacterial cell wall again, and then the different components of the bacterial wall, lipoteichoic acid or peptidoglycan, which can bind toll-like receptor 2, Muramal dipeptide, a breakdown product of the peptidoglycan, which can bind NOD-like receptor 2. And then the surface layer protein itself, which can engage the C-type lectin receptor, DC sign. NOD2 also induces the caspase-1 inflammasome through NLRP1. So these are the different pathways that we know exist just with wild-type acidophilus. And if we look at the big picture of how we can induce a robust immune response, we can see here, the basic categories of toll-like receptors, NOD-like receptors, and then these arrows are pointing out where lactobacillus is driving these pathways. So TLR2, which also is a heterodimer with TLR6, and lactobacillus activates this pathway. And then the muramal dipeptide, which drives the NOD2 pathway, and then the NLRP1, which is driving this caspase. And so, our question was, what's the relative outcome of activation of these different pathways by acidophilus? To assess this, we set up a cold culture system with acidophilus and then bone marrow derived macrophages. And shown here in this picture is the uptake of lactobacillus here, this yellow-green, into the cytoplasm of these macrophages in tissue culture from a wild-type mouse. So just taking these macrophages and then looking at the outcomes, and specifically looking at the macrophage activation here, and then the induction of inflammatory cytokines and chemokines, and then the counter-regulatory cytokine IL-10. And then we can do the same sort of co-culture with mice that have a knockout of the TLR2 gene, NOD2 gene, and caspase-1, each individually. So if we look at TLR2 knockout mice, we can see the only thing that really changes compared to wild type is that we no longer have induction of IL-10. But if we knock out NOD2, we lose all of this. So everything that we saw in the wild type disappears when NOD2 isn't present. So really pointing out that NOD2 is a critical pathway for the immune activation by wild type lactobacillus. And then if we just look at caspase-1 knockout, we still have the maturation of an activation of macrophages and the induction of the chemokine IL-8, but we lose all of these inflammatory cytokines and then the counter-regulatory IL-10. So, this is really interesting. NOD2 is so critical for lactobacillus acidophilus to induce these responses. What's the effect in animals? And we had shown previously that if we just use NOD2 knockout mice, that we do not see a humoral immune response. But then the question was, what cells are critical for NOD2 expression? Because NOD2 is expressed in a number of cell types, including the mucosal epithelium, the intestinal epithelium, and in dendritic cells. So we set up this study using a Cree-Phlox system where the NOD2 gene is floxed, and then we use the promoter CD11C, which is expressed in dendritic cells to drive the Cree recombinase. So these are busy line graphs here showing the endpoint titer for both fecal IgA and serum IgG over time as we've immunized mice. To do a study like this requires a number of controls, but I'll focus your attention on a few key groups. So basically, the positive controls include this dark blue line, the green line here, and then this aqua line here. the key group to focus on is this NOD2 dendritic cell knockout group that was immunized with OVA. So all these boxed groups here were immunized by oral gavage with the OVA recombinant lactobacillus. These others were just given control treatments. And if you look at the results, they're pretty striking. So the positive controls all generate antigen-specific IgA or antigen-specific IgG. But this red line here, the group with the dendritic cells that have NOD2 knocked out, there is no response. Again, just pointing out the critical importance of NOD2. So if we think about that, we may want to expand the innate immune activating profile of a vaccine platform based on acidophilus. And we went about this by, first of all, thinking we would like to add a TLR5 adjuvant. And we chose flagellin, a subcomponent of flagellin, which we expressed on the surface 2 different ways on the surface of lactobacillus. We were interested in this TLR5 ligand because it is known to be important to induce IgA in mucosal immunization strategies in the gut. It also activates NLRC4, which also drives that caspase inflammasome pathway. So, and we can test the biological activity. We see by adding the Fly-C now, this bacteria is activating TLR5. It always activates TLR2. It always activates NOD2. So that was one adjuvant strategy. Another one that we were very interested in is to try to target immune inductive sites. So get the lactobacillus into the immune inductive sites in the intestine where it can really drive an antigen-specific response. So we wanted to target these critical M cells and these are specialized epithelial cells that overlay Peyer's patches in the intestine. And the Peyer's patches are loaded full of dendritic cells, B cells, and T cells, and that's really where the antigen-specific immune response is driven from. And so we did this. We used this fimbrial protein from E. coli, which is known to bind this protein GP2 on the surface of M cells, and we expressed it again on the surface of the bacteria. We can show that indeed the bacteria will bind the GP2, and then when we give this orally to mice, we can see that there is increased trafficking of the bacteria to the mesenteric lymph node, shown here. So this is the FIMH expressing lactobacillus, and now we can actually find these bacteria in the mesenteric lymph node. So we're reaching the Peyer's patch, it's being translocated to the mesenteric lymph node, and then potentially driving a more robust systemic response. And so, you know, what does this look like as we're assessing our adjuvant strategies? And again, we're looking at 2, the Fly-C or the FIMH. We're looking at IgA secreting cells and the Fly-C as compared to this control. And here we're using a really weak peptide antigen that's also co-expressed by the bacteria, Emperor. This is from HIV. It's a horrible antigen. And if you just give it alone, you see virtually no immune response. But if you create a construct with Fly-C and this weak antigen, then you can see really robust induction of antigen-specific IgA secreting cells. And FIMH itself also is driving an increase as well. And then if we look at the serum IgG with the same setup, then the FIMH is driving a robust systemic response, and the Fly-C also very robust on the systemic response. So we're seeing that adjuvant effect that we're seeking here. So now if we back up and look at this same chart with these two adjuvants, we've really expanded our potential to engage the innate immune response and then drive that antigen-specific adaptive response, because we've got what was inherent in the lactobacillus to begin with, TLR2 and 6, and then NOD2 and NLRP1, but we've added TLR5, we've added NLRC4, and then we've targeted the bacteria. So moving on to how are we going to apply this to a coronavirus vaccine? The goal of these vaccines has always been the same, to either prevent or reduce viral replication and to also prevent moderate to severe disease. That's the goal. We know a strength of our strategy here is to induce mucosal IgA because we're going at this with an oral delivery and we're reaching that mucosal immune inductive site. The initial focus has certainly been on spike peptides and spike subunits, and that's really what has been the focus of the vaccines that are currently being deployed. But we are also interested to incorporate other viral antigens so that we have a strong engagement of T cell responses and ultimately trying to drive a durable immune response so we're not having to re-immunize frequently, every year, every couple years, whatever it might be. We were also keen to avoid a dependence on the receptor binding domain of the spike protein. Okay, so this is really a critical point. The current vaccine strategies are very focused on the spike protein and the induction of neutralizing antibodies against the receptor binding domain. And so they are susceptible to immune escape by the variants that might emerge. And just to give an idea of why this is such an issue, this is the spike protein here with all the different domains. And these percentages are looking at the similarity of SARS-2 to the SARS virus that emerged in the early 2000s. So these are very closely related human coronaviruses, but you can see how low the homology is in this region, in this S1 region of the spike protein. So there is just this inherent mutation that's ongoing in this region, and it's a risky area to put all of your chips on as far as a vaccine. So, you know, how do we put this all together and construct a recombinant lactobacillus? So what we have achieved is the expression of two different spike protein peptides shown here, and this is a flow cytometric analysis showing the high level of expression of these spike protein peptides here and here, along with the Fly-C and the FemH all together, and along with a membrane protein that's expressed in the bacterial cytoplasm. We included that because we know that the M protein induces the highest frequencies of T cell responses, and that this may be valuable again because we're inducing a broader immune response and maybe more durable. And this is just a sequence of that membrane protein with highlighting of those T cell epitopes. So we know that this is immunogenic, and we've looked at this in mice, and we see that the peptides, and these are fairly weak antigens, these peptides are stimulating an immune response both in terms of IgA and IgG. And what's interesting about these percentages here is that it aligns very closely to what we're seeing in humans. So using a lactobacillus platform, we can see induction of antigen-specific responses at about the same rate as what's currently observed in humans that are dealing with natural infection by COVID-19. So where do we really want to go with this? First of all, we need to determine whether that S1 region that has that receptor binding domain is actually required for protection. If the answer is yes, then we're going to have an ongoing concern with variants. So we're spending a lot of time trying to understand, can we protect without being completely dependent on this region? So we're also interested to understand, can we drive immune attenuation as opposed to immune escape? So right now, there is a tendency, we're seeing it in populations, for the virus to find ways around the vaccine. But can we target the functional and structural critical areas of the vaccine and drive it to a more attenuated form? And that's why we're using those two spike peptides that I showed in the previous slide and in that construct. So essentially, we're looking at a question of whether we can accelerate this host pathogen coevolution. And then finally, we really have a larger goal here of seeing whether we can develop a lactobacillus vaccine platform that can be used as a pan-coronavirus vaccine. So can we prevent the next coronavirus pandemic by choosing conserved antigens and then deploying this globally, having it available and affordable and effective in low and middle income countries where we can really think about avoiding having to go through this again as a global population. So finally, I just want to point out everybody in my laboratory that's contributed to this work. And again, acknowledge my great collaborator, great friend and mentor, Todd Kleinhammer, who we lost way too soon. And then new collaborators at North Carolina State University, Rudolph Baranju and his laboratory, and Dr. Aganobu Kajikawa at the Tokyo University of Agriculture. All this work has been funded by NIH and Morris Animal Foundation. Thanks so much for your attention. I'm happy to take any questions.
So thank you, Greg, for this very nice presentation. There are, of course, a great number of questions. And maybe that was also the first question raised, is how are these engineered probiotics already allowed to be used in humans? And if not, how long or what do you think are the big hurdles?
Right, it's a great question. So there are recombinant lactic acid bacteria therapeutics that are far along in FDA approval. That's great news. They're doing well and they're entering phase 3. So the FDA has been dealing with this. There's a pathway for these sorts of bacterial-based therapeutics and vaccine to proceed through the approval process. I do think it's very feasible.
Okay, indeed, hopefully. And then we also have a question from Anna Hapfel, one of our students, or actually the chair of our Students and Fellows Association. A very relevant question: how complex do you think this production of these vaccine probiotics is? And it would be really great if it could also be done in low and middle income countries.
Right, and that is the key. That's clearly our goal. So we see that our constructs are stable. We are looking to ensure that we have predictable expression of the multiple antigens that we're inserting into the genome. These are all inserted, they're markerless. So the manufacturing, once it's reduced to practice, it is not that different than cultivating the wild type lactobacillus, which is great. We know how easy it is to grow and inexpensive. Certainly, you have to monitor for quality control of the expression levels of all of these antigens and adjuvants, which adds that monitoring, that QA/QC component to it. But that's why we're excited about this strategy, that it is cheap to do, and it's really relatively simple.
Yeah, that would be great. So actually, unfortunately, it's time. We have to end this Q&A session, but you will receive all questions and there will be also other opportunities for networking. So thank you very much again for this very nice presentation.